Anders, perhaps we should start. It's now almost 5 minutes past 4. Nice to see so many of you in this virtual meeting about the automated patch clamp in CROs. Today we have Mike Morton from Connex who will give a talk about their work in cardiac, and our very own Anders Lindqvist will start off talking a bit about what Sofian does in CiPA and cardiac. So take it away, Anders.
Expectation was that you had the presentation.
Here you go.
Oh, thank you. Thank you. All right. I was going to start off with just some hERG-related stories, or history, I should say. The picture is, of course, Desitin's ECG of a trousseau de pointe, which is published in 1966, which is rather famous now, where you see this typical change in QRS polarity. 3 years after this, Höchst introduced the compound prinolamine. Pronunciation might be different. Anyway, this compound has its fame due to that it was the first compound that was withdrawn for TDP in 1988. This was in the wake of I'm sorry, I hear my own voice here, so it's a little bit strange. Where was I? Yes, prinolamine withdrawal. It was withdrawn in the wake of an awareness that the non-cardiovascular drugs had a teratogenic potential. And this withdrawal was, of course, followed by several rather well-known ones, like teradilin in 91, terfinidin in 97, and astemizole in 99, just to name a few. Mid-1990s also saw a number of seminal hERG papers. Here I only have 2 of them, the Wormke PNAS from 94 and the Sanguinetti from 95. There are, of course, many more that sort of deserves mentioning. From a more safety perspective, we have the foundation of the Safety Pharmacological Society in 2000, and we had the Will Redfern paper published 2003, where he characterized a number of well-known teratogenic compounds in a graded scale. And then in 2005, we have the S7BE14 guidelines published for the first time. And just prior to this, the first automated patch clamp instruments were sort of launched to the market. In 2013, we had the CiPA initiative with the intent that proarrhythmic risk should primarily be assessed using non-clinical human models based on an understanding of the mechanism behind the trousseau de pointe proarrhythmia. This goal might not quite have been met, but it had led to a number of quite important changes in the S7B Q&A, which is updated as we speak. Let's see. So, as said, there are a number of updates to the S7B. There are, of course, I will only touch here upon those that are that concerns ion channel screening directly. There are, of course, more changes in the in vivo part of S7B and also the E14, and all those changes are discussed in the 2 webinars listed at the bottom of the page here. But essentially, up to now, an hERG assay has only been used to inform the first time in man decision point. And after that, the safety pharmacology has essentially started over again, and this hERG data has largely been ignored. In this updated Q&A, which is important, this hERG data, or S7B in vitro core assay, can be used to support clinical development later stage and be a part of the integrated risk assessment, which will now contain both clinical and non-clinical parts. And this can be used to sort of reduce number of clinical trials necessary. It can improve labeling, which is of course very important to the companies, support lower clinical exposure doses needed, and also support alternative clinical study designs. The in vitro part of the S7B now contains best recommendations, which is new. They touch upon things like recording temperature, voltage protocol, test concentrations to be tested, measurement stability, and similar items. And this is for reducing the data variability in the recordings in order to improve the translation of these clinical findings to the clinic so that they can support the clinical development. It also states how to define a safety threshold for a new compound, and also suggests some optional follow-up studies, which can be multiple ion channel measurements, such as looking at the calcium current, sodium late peak currents, which has been sort of been discussed extensively in the context of CiPA. As said, automated patch clamp devices started to appear approximately 20 years ago, and they were sort of since then been an integral part of safety pharmacology departments in order to cope with this increased demand in primarily hERG testing, since that obviously became a bottleneck when this when the S7B was implemented. Over the years, there's been an increased miniaturization, technical improvements, increased throughput, and I hope increased user friendliness of these instruments. And what can be a snapshot of what can be what you can see with these instruments is the paper by Kramer et al. Listed at the bottom of the page, which was conducted in the context of the CiPA initiative. And a question sort of for these devices and the companies behind them is now how to improve support for this these new best practices requirements in the updated Q&A. And one very immediate consequence of this is the development of an open file format for in vitro ion channel data developed by Niels Dybdahl at Sophion and others, which is intended to be used when, for example, filing data to the FDA. And this is a large step forward for ion channel data, or harmonization of communication of ion channel data. However, not everything about hERG is about regulations, dose response curves of a number of compounds or back practices, and there are actually a few surprises still out there. And Dr. Michael Morton of Apconics will now take us on a journey to maybe some more or less traveled corners of the hERG landscape. Please, Mike.
No, thank you. Thank you, Anders. Yeah, that was everything you just said was everything I wanted to say in my first few slides. And I thank you for a great introduction. Thank you everybody for joining today. I hope you're all keeping safe and well. Thank you to Sophion for the invitation to and to speak and to give this webinar. I am Dr. Michael Morton, as introduced, and I'm director and co-founder of a company based at Alderley Park in Cheshire called Apconics, where we have our own ion channel laboratory. I always give my email address in my presentations, and very much welcome any feedback, any comment, or any questions from anyone in the audience as a follow-up to this. I always like to hear from people. So the presentation outline for today, Anders has already given a brief introduction to cardiac ion channel testing, so I won't labour the point there. But as he's alluded to, I'm going to talk about probably a little bit more sort of a bit unusual subjects after that, a little bit more diverse. In the number one, the effect of intracellular pH on hERG inhibition. Number two, a section on cell penetrating peptides. In part three, calcium channel trafficking. And then, of course, what talk would be complete without mention of COVID-19. So the next two slides have really been covered by Anders, but of course, hERG, firstly, as an identification of a molecular target causing torsade de pointe's fatal cardiac arrhythmia, was remarkable in the late 90s, and that knowledge, together with an equally remarkable development of automated patch clamp, moving from a Nobel Prize winning technique in the 80s of conventional patch clamp to something with an industrial throughput, namely ironworks in its in its early days at the turn of the century. So that that and the identification of hERG as the molecular target for drug-induced long QT syndrome, both of those led 15 or 20 years of significant screening campaign, whereby pharma would aim to design out its hERG liability during lead optimization. And the final comment there, that there have been no drug withdrawals since due to this phenomenon. So that's a remarkable success story. And in the electrophysiology, in the cardiac electrophysiology field, that's maybe a little bit of an outdated story now, but still very, very valid, I believe. As Anders stated, where the world has moved on to is the CiPA initiative, which is now 6 or 7 years since its inception, and it has been a great collaborative effort from FDA, academic, pharma, and CROs like ourselves, demanding a more thorough investigation of cardiac ion channels. 7 such channels are listed there. There are, of course, more ion channels in the heart, but those 7 were picked, initially. And in the first instance, this data should be collected by automated patch clamp with a high gigaohm seal resistance, so exemplary quality data, or data quality rather, and that this data should be input into an in silico model to predict cardiac action potential and define proarrhythmic risk. The next pillar of CiPA was that these findings should be verified in human cardiomyocytes, and of course either the company Nanobios on the west coast has found great success in harvesting human material, native human material there, and using that in their models, but equally well the developments in the field of stem cell derived cardiomyocytes in their generation and their provision, but also in the technologies of microelectrode array and voltage sensitive dyes. So again, the field has been furthered by technological advancements, and of course finally, there is a clinical element to the CiPA initiative of replacing the thorough QT study with an earlier phase 1 appraisal. And there are new biomarkers there, JT peak, for instance, of interest, identifying pure hERG block or multi-ion channel block. But that's there are great publications in this area, great presentations. This talk now takes a slightly different approach. Again, a paper that Anders mentioned. There is, significant variability in ion channel data. Always has been. We all have our favorite holding potentials, and we all have our favorite quirks. Every electrophysiologist, and this has demanded, and for the collection of the CiPA data, it has demanded and will demand in future further standardization of the solutions, of the voltage protocols, and aligning platforms. So you would be forgiven for maybe thinking that in the pharma world or the CRO world, that it's all become terribly standardized, and nothing different happens and nothing exciting happens. And my stories today that I want to tell you are some of the the bespoke requests that we receive, and the non-standard stuff, the quirky stuff, if you'll forgive the English, phrasing. And the first is a client that came to us with some unexpected hERG SAR, some activity there. They're an ex-big pharma group, a group of very talented chemists. So know what they know what they expect to see really with their their design of their chemicals. And so they had some unexpected SAR, and then identified a paper that was investigating really the role of intracellular pH, or changes in intracellular pH, and how this might affect hERG potency. So we have the flexibility to look at this, and we're only too pleased to do so. So on the left there is the paper they gave us to have a look at, Wang et al. From 2016, and it is a single standalone paper in the field. This hasn't been investigated a lot. There is one credible paper in this field, and it was looking at difetalide and how, with changing of intracellular pH, difetalide, the onset of block, and also the resulting potency of difetalide clearly changed between pH 7.2 versus more acidic. So based on this, we had a look at difetalide again by automated patch clamp. So we're able to gain significant statistical numbers on this. But clearly you can see in that middle dose response curve, I'd have great difficulty convincing you there's any difference there in the potencies of difetalide, whether at 6.3 or 7.3. And then with respect to the onset of block in the right hand panel, again, you probably couldn't get two more lines, 6.3 and 7.3, overlapping each other. So in our hands, difetalide changing pH had no effect on difetalide inhibition of hERG. So there's one paper that says it should do. In our hands, there is no difference. And subtly, in looking at this, we did find another paper saying extracellular pH certainly has an effect on hERG currents, but also on inhibition by some compounds, but intracellular pH didn't have an effect. So we can only go with the evidence at hand, and in this case, this wasn't for our client. This wasn't what was happening, and we looked to other reasons for a possible disconnect. But certainly, an interesting and concise piece of work, all backed up by good science, and all done by automated patch clamp. The second story to tell you about was during routine screening of a CiPA panel. In this case, one of the first channels offer that we had ready to go was ITO available in culture. So KV 4.3, it's a CHO cell line that we use, and this was recorded automated patch clamp by cumulative dosing, so increasingly from low to high, in gigaohm seal whole cell patch clamp configuration. And you see there on those on the left hand side panel, that's a size of a current metric, and the horizontal lines, the faint lines there are subsequent compound additions. And of those 5 cells, clearly what you can see without too much interpretation needed, clearly there is a point in that cumulative dose response curve that size of current very briefly decreases, but then there is an enormous increase in the size of the current beyond what you would expect to see with a typical agonist, for instance. And it's happening in each cell, give or take within the same concentration range. And when we when we look at this a bit further and expanded, we can see very, very stable currents, and then the signal is just going crazy. The control compound called XC991, by comparison, very stable, and you can see nice concentration dependent increase in ITO block. So clearly something is going on there, with this compound. And so just to having at the same time, automated patch clamp is collecting multiple parameters, not just the old Ironworks days where you had a metric and a seal resistance, that there are multiple, endless parameters being recorded. So we could have a look at that in more detail. And you see certainly, with this compound, early concentrations, gigaohm seal in each and every case, as you would expect in all of these cells. But with increasing concentration, that membrane seal resistance was clearly being compromised, reducing to a point, certainly with our red graph here, where at 3 micromolar, clearly a dramatic and catastrophic drop in seal resistance. Now we do see this with compounds. Our clients might be often are in the oncology space, and of course these oncology compounds by definition might well be unnecessarily so cytotoxic. And so we go back to the client and say, we've seen this. It could be are we looking at cytotoxicity here? At which point our client tells us, oh, actually these are cell penetrating peptides. They form holes in the plasma membrane as they cross. And of course, 2 and 2 exactly explains what's going on here with a disruption of the plasma membrane structure and function, and a collapse in seal resistance. And we can see here on this next slide, again, another parameter that is just routinely measured, membrane capacitance, is devastated, is just collapsed as the membrane breaks down. So that speaks to the relationship with the client being very important. But this obviously, as then a piece of work, we've clearly identified something interesting here. We have identified, something interesting for the client to investigate. And, you know, I can develop sort of, you know, part of the story here was, you know, the client coming back with, well, we've got hundreds of these compounds, you know, can we have them all tested? And, you know, and try and develop some SAR around that. Clearly, the cell penetration is important to efficacy of this target, but can it be done in such a way, a less harmful way, I guess. And so, you know, we started looking at this in more detail. We had a variety of compounds from the client. 10453 there is the molecule we've looked at so far that causes this calamitous drop in membrane integrity. 10453 star is a different compound actually, that the client at the time thinks will lack this activity. And so, you know, we had a little bit of a play with the conditions with our concentration response curve, you know, measuring, adding compound, but then a wash step, and then adding a higher concentration wash step, and so on. And so here at this lower concentrations, we're seeing no loss of membrane integrity. But then as we get to this magic 3 micromolar, in those 4 panels you see there, that is all 10453, we see this, you know, calamitous drop in membrane, in seal resistance, which speaks to the drop in membrane integrity. The wash step does show some reversibility, and it might lead you to think that if we washed more aggressively, washed more often, washed for longer, you know, we might see some recovery of the seal resistance. It probably depends on how low the seal resistance went in the first place. But what was also clear is that the 10453 star, just those 3 panels on the bottom left, considerably less effect on seal resistance at 3 micromolar. So there is clearly some, you know, structure activity relationships here between different compounds having a different effect. And of course, we were very happy to, you know, explore this a little bit further, speaking to the you know, the power and the throughput that we can achieve with automated patch clamp. We were told by the client they have an academic collaboration, and the collaborator had done some work on the effect of, you know, calcium, barium, for instance, on these phenomena. And certainly, you know, we can see on the top 4 recordings there, in the absence of calcium, the compound, sorry, the compound names are changing there, aren't they? This ST101 has a instantaneous, almost within 2 sweeps, which would be all of, you know, 5 or 10 seconds, an instantaneous collapse and complete collapse in membrane resistance, where the 10453 star, or 263, as it's labeled on this slide, you know, there was a more dramatic effect of that compound, but different to ST101. So there's you know, there's interesting you know, SAR to develop there, and when we have that story, you know, complete, you know, we'll be able to share that in more detail as that's revealed to us. But what was the first you know, necessary stage in this was to screen, you know, this you know, modest number of compounds that the client had, and to give them some, you know, with 10453, are there compounds that are more aggressive or less aggressive? We did have a look at this testing at a single test concentration, 3 micromolar, for instance. But there was, you know, there was a lot of variability across the plate, and that wasn't going to be a working assay. But if we took that and did the, you know, the simplest of concentration response curves, taking that, you know, sort of inherent variability, you know, maybe about around that point, but putting that in a context of a concentration response curve, gave us much more, equivalent data, and you can see a certain uniformity across the plate here. Left-hand side going from high to low, right-hand side going from low to high, or the other way around, just to see are there any, with automated patch clamp, are you looking at any pipetting differences or timing differences? So yeah, we had the makings of a very simple and straightforward assay there, where we could probe the compounds with reasonable throughput and get reasonable data that the client could apply to some structure activity. So just by example, just for example there, 2 runs that were completed. What was decided, because of an inherent variability on one hand, but also really just good science on the other, to have this 10453 as an internal control on every plate. And then X number of other compounds were tested, and you can see quite clearly some compounds do have a different profile, and some compounds there, clearly the y-axis there is the percentage of cells losing seal resistance. There are compounds there that are less aggressive. But there's one compound there on the right-hand panel that shows very little, cytotoxicity. So it's giving the client something to work with there, and that's even ahead of at some point this client will, in fact, has already, they have in screening all of these compounds developed an SAR. They will be able to match efficacy to a better profile in automated patch clamp, or a less aggressive effect on seal resistance, and then we go back to CiPA and getting some cardiac safety data, not forgetting that that's always our desire. But certainly an interesting story that took us away from the routine into a nice little bit of work, nice bit of science. That's when we tie all this together in a complete story with the client, I think it'll be a very an interesting story. So just moving on to another a story here, number 3 of calcium channel trafficking. hERG trafficking has is a thing. It is something we do, and rightly so. Why should drugs just have an acute 3-minute, 5-minute effect on an ion channel? The classical compound here, the gold standard, really was pentamidine, a compound that was shown not to necessarily have an acute effect on the hERG potassium channel, but did cause ECG changes, and the result that was a result of a long-term change in the expression of hERG, the actual protein itself at the membrane. And so so it was right, therefore, that some compounds, for maybe an unexplained QT prolongation, that we should measure expression of the ion channel, and we do this for hERG, and our in-house protocol is based on a communication from Yoshinaga, where compounds are added for a more chronic period, 24 or 48 hours. Across a 384 well platform, we can generate big numbers and have a look at the absolute levels of hERG current, and this speaks to then changes in hERG expression. Pentamidine response curve is given there on the left-hand panel, and another drug that happens to do this is fluoxetine, and we can see there the change in expression with fluoxetine. There is a caveat to studying trafficking this way, is that it's rather stating the obvious, but the compound cannot be a potent blocker of acute blocker of hERG potassium channels, otherwise we see the same reduction in hERG expression, in the hERG current. However, we can measure that, and certainly, pentamidine and fluoxetine is certainly more potent at the trafficking. So that, that's hERG trafficking. But if we're interested in hERG trafficking, why aren't we looking at all the other, CiPA ion channels for trafficking defects there? And so a client, asked us to take a look. They had reason to, ask about a calcium channel trafficking assay. And the only channel here, the only issue here is trying to grow enough calcium channel cells to actually, do the work. But you'll see there, on the y-axis in that left-hand panel, a calcium channel expression. It's obviously an inward current, so it's negative numbers for big expression. And on the bottom there, the compound is just simply numbered, compound number 29, compound number 30, 31. Ignore the rest of they were just various controls and stuff. But clearly it doesn't take too much interpretation there that with compound 29, particularly approaching 10 and 30 micromolar there, clear increase in the calcium channel current, and this wasn't an acute agonism as we would see with a BK compound or FPL. This was a change in, it wasn't that we measured that, and this was a change more chronically after 48 hours of incubation of the compound with a CAV 1.2 cell line. So, in this case, we can report that back to the client. Of course, science 101, we have no control, we have no control here, negative or positive. So, the finding is what it is. It's one of those experiments, we're happy to do, but there is, at risk that the data, if we see nothing, that's largely uninterpretable. But yeah, it's the data was reproducible, and certainly the client was delighted with the work that we'd done. Just as as a student project in the lab, when we were working on on hERG trafficking assay, it's just interesting. If we're looking at one channel, what about the others, and does pentamidine inhibit trafficking of the sodium channel? It's known that pentamidine binds hERG at a conserved phenylalanine residue 656. There was no effect, in our hands of pentamidine on the sodium channel trafficking up to 100 micromolar, as measured similarly by a functional readout. And it does so happen that the sodium channel lacks this conserved phenylalanine. So mechanistically we can maybe understand why that is so. But an interesting story nonetheless. And certainly, with the hERG trafficking and the calcium channel trafficking, it all speaks to an integrated approach to in vitro, cardiac safety liability. So just a final slide, and just a throwaway slide, really, in these challenging times. What about COVID and ion channels? And I was commenting with Anders the other day. People have been studying this for years, and I would suggest probably without much funding until last year. I guess when everyone was suddenly hugely interested. But it is apparent that some of the viral proteins do induce ion channel currents in the host cell. And that these ion channel currents are certainly sensitive to a amiloride derivative, and the protein 3A showed some potassium channel selectivity. It's difficult to prove something is an ion channel or not, or whether it's a chaperone, but certainly it's not without precedent that certainly influenza M2 and the HIV proteins and some of the proteins from those even small genomes can modulate host ion channel function. So it wouldn't be unreasonable to expect there may be something there with COVID, and terribly exciting, I'm sure, for the the people on the call who live in the ion channel world, that this was the case. But of course There is the the need for evidence of bona fide ion channel behavior, and then this opens the world to what are the antiviral effects of possible therapeutics of modulating these these COVID channels. So it is an exciting area, and I did want to mention that to you today. That concludes my presentation. Certainly would love to hear from you. Thank you for your attention, and I think we're back to Damien now, who has managed to join us. So over to you, Damien.
You all hear me now? Hooray!
Yay!
Maybe you actually preferred it without me and my ugly mug on the screen, but you've got me, unfortunately, so stick with it. Great talk, Mike. Thank you, and thanks to Anders as well for the really interesting and useful intro too. We have got a couple of questions, Mike.
Yeah.
I'm going to go to you initially, and if I just go back to my chat. So, Sarah Lilly, hello Sarah, nice to sort of see you logging in as well. What level of agreement is being seen between in silico prediction or modelling and cardiomyocyte studies using planar patch clamp?
Yeah, no, thanks. Thanks, Sarah, and hi, and thank you for the thank you for the question. The data is very much being collected, and is gathered. Is some published, and some more will be published. We in our hands, we have certainly found that there's certainly great value in in the in silico modeling, that if you saw in front of you a mixed ion channel block, in your mind, trying to put that into context could be challenging. So we've had some powerful examples where the in silico modeling has given great context to that data. So certainly incredibly useful. And yes, there's always cases, often depending on concentration, etc., where these things, may or may not tie up. But there's, I think there's a lot of evidence for, reasonable translation, with any number of caveats. But I think CiPA as a whole, I think I expect, we're entering many years of data collection and learning, and I think certainly the FDA is inviting that. I've no doubt for big pharma is going to collect all of the data and address exactly these questions and learn from years of data sets how this is going to play out. And as the as the CiPA working groups gather more data on the reference compounds, these will all be modeled and go through the stem cells as well, so it will end up published as well.
As an almost follow-up to that, Mike, I mean, obviously you highlighted in the Kramer publication from 2020 that there was a degree of variation between different platforms and different protocols, and something that CiPA is obviously trying to standardize. And even with standard protocols, there's still a degree of variation there. I mean, with that in mind, and almost sort of, as I say, as a follow-up to your answer to Sarah's question, where do you almost sort of see CiPA going in the future? I mean, do you think we'll get to that point of having a standard protocol that we can all apply using APC to, the numerous different sort of channels, or do you think that's still a bit of a, I don't know, a way off?
Yeah, yes. Thanks for asking that one. I mean, what CiPA, this data collection, the hERG story took 15 years to be adopted, and there are still medicinal chemists out there that will give you a hard time about hERG. So it's got for acceptance and to universally be accepted, and and I guess there's going to be a long period of data collection, and then hopefully the case studies will come through that actually look what we found, and look how it translated. And everybody deserves that success. As an electrophysiologist, first and foremost, anything raises interest and, you know, puts ion channels in more in focus, you know, is great, and rightly, you know, rightly so. You know, they are critically important, and you know, the philosophy of, you know, just doing, you know, hERG at the end of discovery for your first time in man package, you know, no, there's more to do here. And so if, you know, if it's raised that awareness, then fantastic, and fantastic for all of us in the field. The standardization, yeah, is criminally difficult. I'm glad you actually didn't ask me how to standardize all of this. you know, because it, you know, it isn't, you know, it almost feels like a one size doesn't fit all for a voltage protocol. And then how do you address sodium channel use dependence but keep calcium channels from running down? You know, that can't be done in the same protocol. You know, please, yeah, debate. But yeah, I guess, you know, and Kramer's done a great job. Not for the first time. Kramer's done a fantastic, you know, seminal paper. You know, but where are the differences, you know, coming from? You know, there is something here about compound solubility and testing to steady state and glassware, and you know, getting some of the basics right. I am not saying anyone isn't getting the basics right. I'm not saying that, but those, you know, this was all, you know, in the hERG field, Ironworks, you know, gave a potency of 20 micromolar, and then suddenly at GLP it's 1 micromolar, and everyone is screaming. And you know, this has been studied. You know, it's plastics, it's compound solubility, and you know, it's and I think these things are important, and you know, maybe that's where it sits, because great efforts, huge efforts have been made to standardize, you know, a lot.
Yeah, I think you're right on both counts, actually. I mean, there's a degree of, you know, there's so many different variables, as you say, right down to glassware, plastic wear, solubility, all of these tiny variables that we know are all potentially affecting the differences, and why some, even with the sort of standardized protocol, it's still not quite working. And yet, getting back to your first part of that answer, the ability for us to sort of get large amounts of data sets with high throughput APC means that with those data collections, we should be able to get sufficient data to start narrowing down all of these different elements, even though there are multitude of different elements that are affecting clearly even our supposedly standard protocols. I do think those two together, whereby you've got high throughput, you've got large data sets that can be generated, and you'll hopefully be able to sort of narrow down, as I say, filter down to some of those key elements that we do all need to be aware of and standardizing in our protocols. Anyway, I'll move on. Maybe a question I can put to Anas is from Shankar: can we record cardiac action potential using automated patch clamp?
Yeah, that's a good one too. Well, yes and no, of course. I mean, in my hands, in my very personal experience, the success rate using IPS-derived cardiomyocyte has been, let's call it limited. I think that's a fair expression in English. And that is one point, that we need to sort of boost the success rate, and we are putting quite a lot of effort into it right now. The other one is, of course, that most instruments are using standard wholesale recordings, which means that you sort of to some extent lock the membrane potential by the solutions you use, and you will get the corresponding answer. And if you have a intracellular extracellular solution that sort of depolarizes the cells just due to the nature, then you will have an action potential accordingly. So I think if you want to get sort of some kind of realistic value of the membrane potential of a cell, you probably have to go to a perforated patch and of some kind, and we're putting some effort into that as well, but unfortunately I don't have any results I can share with you, so just from top of my head.
Yeah, that was a bit of a curveball I threw out to you, Alice, but thanks for sort of making a really good stab at that. I mean, bottom line is, Shankar, that even manual patch clamp recordings on cardiomyocytes are really challenging, because you've basically got this long tubular cell that's often contractile in nature, so it's potentially even moving while you're trying to patch it, which, ask any patch clamper, and that's, you know, a disaster waiting to happen. So even manually, they're not easy to do. And yet we are making progress on automated patch clamp with, for instance, induced pluripotent stem cells derived cardiomyocytes. These are more amenable to automated patch clamp, and with things like the current clamp that our amplifiers and many platform amplifiers can do now, just like a manual patch clamp can switch between voltage clamp and current clamp, so can an automated patch clamp amplifier, which means that you can make, if you are sufficiently successful with your recordings against, for instance, a cardiomyocyte derived from a pluripotent stem cell, you can make those current clamp recordings to potentially record action potential like recordings. So it's not easy, but we are making significant progress in that field. Another question, I think, for Mike. So Batu is asking, on the cell penetrating compounds, are they possibly disturbing the pH? What might be a possible strategy to overcome such gigaseal problem in such cell penetrating compounds? And the third part of that question, do you think such compounds are still drug candidates to modulate the channel?
Yeah, no, thanks, Batu. Yeah, you know, yes, cellular pH, you know, and its control is, you know, a necessary part of that is an intact membrane. You know, yeah, that's you know, that's homeostasis. And yes, so anything, yes, so cell penetrating compounds, you know, anything punching a hole or compromising the membrane integrity, you know, yes, could be you know, changing intracellular pH. I can't sort of speak to that. Yeah, you know, I can think of a few experiments on the back of that I would like to you know, try with an acid sensing ion channel, you know, maybe, and then throw some of these peptides out there. But yeah, you know, certainly it feels like entirely plausible that it you know, could happen. You know, the possible strategy to overcome this, I'm going to leave in this case to the client. You know, this work is ongoing. You know, the client knows all of the you know, the structures and the IP around charge and the length of the peptide and the linkers, etc. And you know, I'm going to leave that to them. And then, you know, when all of this work does fully read out, I hope we'll have a, you know, a better and fuller presentation with structures and much more understanding of the SAR. I've given you a very sort of a you know, well, less than half the story here today. My hope is it will develop and we'll have a much better story. Do I think they're still, you know, capable of drug candidates? You know, yes, you know, absolutely, absolutely, I do, yes.
And Mike,
stay tuned.
Mike, I'll have a follow up on that as well. This is the great thing about being host. I can just keep peppering you with questions and put you under the spotlight and grill you a bit. But it's, you know, it's purely based on my curiosity. You understand, of course. Those oncocytotoxic compounds, and apologies if I missed this, I may have missed it, but it's very striking, you know, those sort of, I think it was five cell recordings that you sort of showed, one on top of another on the left-hand side of your screen, or left-hand side of the slide. And it's very striking that almost every one of them started to have this degradation in the seal resistance at a similar time. Did you see that time dependence being a concentration dependent effect? So very high concentrations would do it far faster in terms of the kinetics of developing that sort of state of breakdown.
Yes, certainly, if you went straight in there, 10 micromolar, it happened much more quickly. It very quickly became apparent there were, suddenly a million experiments we could do to chart this. And, maybe, the academic collaborator, will pick that up and, and of course, on a, APC with all the throughput, you could rack through those experiments quite quickly. There were questions raised, about the ion, is it in any way dependent on the ion channel function? And what if you start blocking that ion channel function, etc. So, there were some fascinating questions, certainly around the divalence as well. Where, but there's an element of, sort of research there. Where we wanted to get to, and the client wanted us to get to, is okay. So, but how can we screen in a sensible format a large number of compounds where we can get some SAR data? So really, it was trying to ignoring the million tangents and actually, okay, but what does a more medium throughput assay look like, where we can just scan across a selection of compounds? That's really where that, we could have done a million experiments that would have been, fascinating. And
one final question. So this is your call. If anyone wants more questions, you've got to start typing them in the chat box. But one final question from me, Mike. I know we're all quite busy, so I don't want to keep you too long.
No, not at all.
But I think that point at the very end there, where you were talking about calcium channel trafficking, is a really salient and pertinent one, as in, we often concentrate, as we did in the very early years of all this cardiac safety ion channel assessment pharmacology, we often concentrate on the bad boy of cardiac ion channels, the hERG. And so, people have done hERG trafficking studies, but there's been very little done, as you're suggesting, against some of the others. I mean, in that sort of idea, I guess part of CiPA almost needs to be, because we're often looking at the acute effects on ion channels, probably in the tens of minutes, maybe into an hour of exposure in sort of extreme cases in a lot of our work. So effectively very acute exposures. We know that these compounds are going to be sticking around in the body for weeks, sometimes months in some cases. That they deliberately have been designed in that way to be, long lasting and persistent in some cases. With that in mind, I mean, should we be doing more chronic exposures and determining these kinds of potential trafficking events and modulation?
I guess, one answer to that is that's what's been afforded us by the stem cell working, group, is that, that technique and that technology, affords an incubation for 24 or 48 hours, maybe longer.
Yeah.
And then measuring, the action potential, that way. That's one answer. Yeah, no, you're right. It's very rare for us to take a drug for 3 minutes. it's very, very rare. So yeah, it is. But I guess if we, but right now is measuring for the 3 to 5 to 7 minutes, actually capturing much of the liability. Really. We have the stem cells to catch more, and then for now, let's get that right. And in the and for now, everything else, will be later in the safety profiling, will be apparent. For now, yeah, how you would study, these phenomenon purely with ion channels for weeks on end. Yeah, I have to think about that one.
Yeah, I think that's probably right. I mean, as you say, a lot of that work is being picked up in the stem cell work, where you're doing more chronic exposures and seeing morphological and other sort of changes, biomarker changes in stem cell sort of developments over longer exposures. Anyway, we'll call it a day now, because I know everyone's busy, and particularly you and Anas, you need to sort of get back to your own day jobs away from being the bright lights or presentations of ion channels. But great talks, both of you. Thank you very much. And thanks to everyone for their time logging in. It's been great sort of having your feedback and questions. And look out for future webinars. Probably sometime the end of April, I guess, will be our next Sophian webinar. So keep an eye out for that too. But thanks to everyone, and goodbye.
Well, thanks, Damien. Thanks, everybody.